Hello, welcome to Molecular Insights NG. And in today's session, we'll be carrying out a basic technique which is known as gel electrophoresis. Now, the gel electrophoresis will be conducted on samples that are here, that are lined here in this rack. And the samples that are here are gotten from 12 gram negative bacteria. So the DNA extraction method that was used to extract this sample is phenol chloroform method of extraction. It is a very well-known DNA extraction method. And what we also have here is an ethidium bromide that will be added when preparing the agar gel. Then here in this little appendix tube is our loading dye that will also be used during gel electrophoresis. Then we have here is the DNA ladder that will be used as a reference marker when visualizing DNA bands. So the gel electrophoresis is a well-known technique. What is used basically is to separate DNA fragments from the heavier DNA fragments to less heavier DNA fragments. And the fact about gel electrophoresis is that the smaller DNA fragments travel faster. It means that after gel electrophoresis, when we look at using our gel imager, we will see that some DNA fragments move further away from the DNA well. It means those fragments that move further are smaller in size, and those who are closer to the well, the DNA fragments that are closer to the well are heavier. So let's get to work. Okay. For the preparation of the agar gel, what you need is male conical flax. Then you need an agarose gel powder, and the agarose gel powder, the number of grams, the 3 grams of agarose gel powder, which are individually currently here. Then you are going to add the 3 grams of agarose gel powder gently into the conical flask that houses or contains 300 ml of distilled water. Then you take a piece of aluminum foil, paper, and you gently and properly clear the opening of the conical flask. And this will be placed into our water bags. But one thing you should know, before placing it, you have to put some little perforation here at the top. So I'll be doing that using this to create some little perforations on top, create some opening on top. Then this is enough, and this will be placed into the water. why we are placing the agarose gel in water bath, so that the ground gel powder that has been added into the 200 ml of distilled water will get dissolved using the water bath. So that's the essence. That the water bath on, and I will leave it until it gets dissolved. So okay, so after bringing out your agarose gel from the water plant, you allow it to cool to a certain temperature that you can easily feel your hand that it's a bit cool. So what you do next is to add 15 microliter of ethidium bromide to it. There are two ways of doing this. We could have done this earlier at the initial stage by adding the ethidium bromide to it when it was not added into the water bath. But this is the second approach. After placing the agarose gel powder in the water bath and removing it to allowing it to cool, and since it's still warm and it has not solidified, we are going to add 15 microliter of ethidium bromide.
Is it that this is fish?
So we allow it to mix with the agarose powder by gently swirling it. As you can see, this the Tedium bromide is has fully dissolved into the agarose solution. Then you place it back and leave it for some few minutes. Pouring agarose gel into the carbon for polymerization. You first ensure that you have used a masking tape or a sellotape, a transparent one, to seal the sides. Make sure they are properly sealed. Then you place it on a flat surface. Yes. Then you pour. Then you allow it to polymerize. I'd like to see something.
Okay, so here's our gel tank and the DNA rightly seated in the wells. And as you can see, they are migrating toward the positive electrode. The DNAs are negatively charged, and so they are migrating right now. So we'll leave it for 30 minutes and see the outcome.